E as E. coli. Blender cup as microcentrifuge tube. Blender machine as microcentrifuge machine and vortex machine. Transparent glass as beaker. Salt water as lysis buffer. Water as proteinase K. Pot as water bath. Tube as spin column. Syringe as pipette. Sugar water as RNase. And lastly, measuring cup. First, 50 milliliters of coffee was placed into the blender cup. Second, the coffee was blended for 1 minute using a blender machine.
Pallet and supernatant are differ in terms of structure and density. Pallet is a concentrated precipitate that accumulates under supernatant, while supernatant is a liquid that lies above the pallet after centrifugation process. In terms of density, pallet is denser than supernatant. That is why we can see supernatant above whereas pallet below of each other.
Third step. After 1 minute, the supernatant element was discarded by pouring it into a transparent glass. Fourth, 10 milliliters of salt water was added into the blender cup containing the pallet.
During lysis step, the nucleus and the cell are broken open and then releasing DNA. Enzymes and buffers like proteinase K, RNase A, binding buffer, and lysis buffer are used in this step to dissolve cellular proteins and extract DNA.
Fifth, the salt water and the pallet were mixed using a blender machine.
In binding step, the spin column was centrifuged to trap DNA into the spin column matrix, and all cell walls and debris will flow through into the collection tube.
6. 5 milliliters of water was added into the blender cup,
while proteinase K was added to degrade proteins. This is because during DNA extraction, there will be a lot of protein contamination. This proteinase K is stable over a wide pH range and suitable for DNA extraction process.
7. The blender cup was placed in the boiling pot for 5 minutes. 8. 2 milliliters of sugar water was added into the blender cup using syringe.
RNase A is used to degrade the RNA from DNA samples because for this experiment we only use the DNA.
9th, the blender cup was placed on the blender machine and was blended for 1 minute. 10th, the blender cup was left at room temperature for 2 minutes. 11th, 1 milliliters of water was added and the mixture was transferred into tube.
Spin column consists of spin column matrix and collection tube. Spin column is put above the collection tube and used to trap DNA, while collection tube is a tube that is used as a collection vessels for eluted acids during centrifugation process.
12. The tube was shook for 30 seconds. 13. The collected liquid was discarded into the empty glass. 14. Step 12 and 3 were repeated 3 times by adding 0.5 milliliters water into the tube each time before step 12 was repeated.
In cleaning step, the wash and clean buffer were added separately. In these steps, the salt and other contaminants will be removed and left only the high purity of DNA sample.
15. The tube was shaken for 1 minute. And the last step, the tube was kept at refrigerator.
Lastly, the elution step. In this step, elution buffer was added to separate and extract a material from another. In this case, we want to extract the high purity of DNA samples so that it can be tested easily.